Hi, my name's Amanda, and this summer I'm mentored by William Chapman to work on this project about information flow in neural populations. So I'm in Department 1421, which is all about cognitive and emerging computing, and a major thrust in this department is neuromorphic computing. So what that means is computers that operate roughly based on how we think the brain performs computations. But in order to do that, we first need to understand how these brains actually perform computations. So that's the major question of this project is how do local hierarchical computations happen in biological brains? So if you imagine two brain areas, X and Y, each of these are composed of multiple neurons. And in the context of this project, the two levels of computation of interest to us are the neuron level computation and the population level. So each of these blue dots is a neuron, you can think, and then the population is the whole aggregate of them together, like a brain area. So just to give you a sense of the kind of data I'm working with, this is an example of spiking activity in 40 neurons. So each row is a different neuron, and each of the black dots indicates that a spike or some activity was observed at that time point. So the scientific question that I set out to answer in this project is how does information flow between two brain regions during navigation? And for this particular data set, there was a mouse navigating an environment that was either familiar to it or novel to it. And during this time, we're getting simultaneous recordings from multiple neurons in two brain regions. So the approach that I took here to calculate population information flow was simply to take the aggregate of pairwise information flows. So this measure I used directed information. Um, I calculated that for each pair of neurons in the two different areas. And the application of this method to this data set reveals a few interesting things. The first is the relative magnitudes of each of the curves. So blue is information flow from X to Y, and then orange is information flow from Y to X. And you can see that blue is consistently higher than orange, and this is good because it's corroborated by anatomical evidence of how we know these two brain areas are connected to each other, biased towards X to Y connectivity. The next interesting thing to notice is that at the onset of the familiar environment, there's a lot of information flow which then tapers out, possibly due to lack of attention as the animal runs through multiple iterations of the same track. But then once it moves into the novel environment, there's a big spike and ramp up in information flow, which suggests that there could be relevant computations happening due to remapping of the environment in the two brain regions. So this is great, but there's still a computational question of whether this measure that we're using to approximate population level information flow is like aggregates of pairwise information flow is actually a good low dimensional representation. And the answer is not in all cases, because imagine if you had the case where three neurons had to be co-activated at once to produce activity in another neuron. This kind of metric wouldn't capture that because it's only pairs. It wouldn't be like three-way dependence. So that's currently what I'm working on is how do you find these joint clusters of activity that you can define to be units of computation at a population level. So thank you. And I would like to thank also URA and my mentor, William, and my manager, Shradeep.